And this is our defense for Jamileh's thesis. Jamileh, again, I'm pretty certain every one of you knows Jamileh really well, a hyperachiever within our MES program. Very, very charismatic. And also she started the program in 2018, in fall 2018. She comes from USC. She graduated from USC in the Marine Science program, the USC in Columbia, not Beaufort. But she did work with Dr. Montie prior to coming here, so she already has some connections. And I think I met Jamileh first time in my pollution class, pollution and environment, and she again was pretty good. And that's where I got acquainted with her research program with Dr. Montie and got to learn about her research interests, and we chatted a little bit, and eventually I ended up being her research advisor, academic advisor, but she continued working with Dr. Montie during this time. So I'll let Dr. Montie talk a little bit about Jamileh's research as well.
Okay, thanks. Thanks, Vijay. So I'm Jamileh's research advisor, and I'm an associate professor in the Department of Natural Sciences and Marine Biology at University of South Carolina in Beaufort. But I also serve as a faculty in the graduate program in the College of Charleston in Marine Biology and Environmental and Sustainable Studies at the College of Charleston. And my program focuses on acoustic communication, soundscape ecology, environmental toxicology, and diversity and abundance of fish and marine mammals and estuarine systems. So Jamileh, she joined our lab as an intern in 2017. I think it was January, right? January 2017. Just a great worker and really passionate for environmental science, and started working on various projects, including snapping shrimp. And then she joined the program in fall 2018, and her master's project is focusing on understanding factors that influence long-term salinity in fecal coliform patterns and how they've significantly changed over the past 20 years in estuaries of Beaufort County, South Carolina. The project was funded through a vested interest from the town of Bluffton, Beaufort County, and the Spring Island Trust. So the results of this research will inform policy decisions in our county, and we look forward to that. As you know, her committee members include Dr. Vijay Vulava in the Department of Geology and Environmental Geosciences, and a member of the graduate faculty at the College of Charleston. And his research interests are in the broad area of environmental pollution. Dr. Matthew Nowlin, an associate professor of political science at the College of Charleston, and his research and teaching focuses on environmental policy and politics. Kim Jones, she's also on Jamileh's committee, and she works for the town of Bluffton's Watershed Management Division manager. She's the director, and her primary responsibilities include developing and implementing policies, programs, and projects related to stormwater and water quality management. So with that, I'll give Jamileh the floor and she can get going.
Thank you, Dr. Montie and Dr. Vulava, for those introductions. I really appreciate it. And hello, everyone, and thank you so much for attending my thesis defense, reviewing my research titled A Historical Analysis of Salinity in Fecal Coliform in the Estuaries of Beaufort County, South Carolina. In this presentation, I'll provide background information on the importance of monitoring salinity in fecal coliform to assess estuarine health. I'll then review the first portion of my thesis titled A Changing Estuary, Understanding Historical Patterns in Salinity and Fecal Coliform in the May River, South Carolina, and a second portion titled A Broader Understanding, Investigating Changes in Salinity and Fecal Coliform over the last 2 decades in the Estuaries of Beaufort County, South Carolina. I'll then finish this presentation by reviewing current policy efforts and providing key recommendations and insight and providing policy recommendations and summarizing key insights, as well as providing the next steps for this research. So why monitor estuarine health? Coastal development is a prevalent threat facing many estuarine environments in the Southeast United States. South Carolina is not immune to this development, and much of its coastline is comprised of tidal rivers and high salinity estuaries. This development risks harming the ecologically and economically important ecosystems that exist in South Carolina. And so in this study, we utilize salinity and fecal coliform as a means of assessing this estuarine health. And just to understand this kind of development that's occurred within the region, the population of the town of Bluffton is shown in this figure on the left of the screen. Salinity is an important ecological driver within estuarine habitats. It affects the biological processes occurring in many species within the estuary, such as osmoregulation and metabolic processes. Additionally, it also impacts species distribution throughout the estuary, with fluctuations in salinity potentially disrupting this distribution. Through long-term monitoring, we can better understand the changes in the estuary over time, which can impact estuarine species. Fecal coliform bacteria are utilized as something called a fecal indicator bacteria, or FIB, which help provide insights into the extent of fecal pollution in an estuary. These bacteria are primarily found within the GI tract of warm-blooded animals. FIB are utilized as a means of assessing not only fecal pollution within an estuary, but are used as a proxy for more harmful fecal-borne pathogens, which can be viral, bacterial, or protozoan in nature, and are not as easily assessed within laboratory settings. Examples of this being Vibrio and Giardia. A large amount of literature and research indicate that FIB are linked to watershed development and population growth. So as pervious surfaces are converted to impervious surfaces, the amount of fecal pollution that enters the estuary increases. Therefore, in light of the increased amount of coastal development taking place within Beaufort County, it is important to monitor long-term fecal coliform trends. So for my thesis, I have 4 main objectives. The first was to determine the factors that influence historical salinity and fecal coliform trends at South Carolina Department of Health and Environmental Control shellfish monitoring stations located along the May River from 1999 to 2017. We then wanted to compare these patterns of salinity and fecal coliform among the shellfish monitoring stations located within SCDHEC areas 15, 16B, 17, 18, 19, and 20 to better understand which locations have undergone the most degradation from 1999 until 2019. We then ranked the bodies of water from most impaired to least impaired, and we then are publicizing the research in ways that gain both the public attention and the attention of all affected municipalities in order to inform management decisions and assist with the prioritization of remediation efforts. So this research utilized the South Carolina Department of Health and Environmental Control's salinity and fecal coliform data for the past 20 years. Through their shellfish monitoring program, data are collected on a monthly basis by the department. Monthly sampling dates, times, and tidal phases are selected at random. Salinity is measured during the sampling or in the field using a refractometer, while fecal coliform levels are determined in the lab using an A1M 10-tube decimal dilution method, which results in something called a most probable number, or MPN. This is a statistically derived means of estimating the number of bacteria per unit of water. And so this the A1M 10-tube decimal dilution method uses serial dilutions to determine the number of bacteria in the sample, with the number of positive dilutions helping calculate the number. So now I'll review the first portion of my thesis. I'm really excited to note that this chapter has been published in the Marine Pollution Bulletin, and the article can be found online, or I can send you a copy if you request after the presentation. So for this analysis, we used data from 11 SCDHEC shellfish monitoring stations located along the May River, South Carolina, which is right here on the screen. The stations denoted in blue were stations that were sampled from 1999 until 2017, while those denoted in red were those that were added in 2009. The May River itself is a large subtidal river estuary that is approximately 22 kilometers long and 0.01 kilometers wide near the headwaters, while it's 1 kilometer wide near the mouth. So this is the mouth of the estuary, and these are the headwaters. The water depth near the headwaters range from approximately 3 to 7 meters, while near the mouth it ranges from approximately 4 to 18 meters. Bordering the river and creeks are intermittent oyster rubble and live oyster reefs in vast areas of salt marsh that is composed of smooth cordgrass. The estuary is strongly influenced by 2.5 to 3 meter semi-diurnal tides. It's important to note that in 2009, SCDHEC restricted harvesting to oysters in the upper portions of the May River estuary due to elevated levels of fecal coliform. So this led to the addition of these 4 stations in the headwaters to help monitor it closer. Looking at this map, we can see land use and land cover around the May River, which is in dark blue. So this is the May River estuary right here, denoted in dark blue. Surrounded by it is residential in pink, as well as forested lands in dark green and wetlands in light blue. The May River watershed has undergone a large transformation in land use and land cover over the past 20 years. So this figure is 1992. In 1992, the headwaters were surrounded primarily by these wetlands and forested land. And then in 2016, which is this figure, we can see that the forested land has decreased and there's a large increase in this residential land. And so from 1992 to 2016, the amount of residential land use has increased by approximately 13%, while the amount of forested land has decreased by 11.5%. And this trend aligns with the amount of population growth that has occurred in the region over the past 20 years. The town of Bluffton, which surrounds the May River watershed, has increased from approximately 800 residents in 1990 to 21,000 residents in 2017. For the statistical analysis of this long-term data set, we use general linear modeling to determine if certain factors influence salinity and fecal coliform levels in the May River. Additionally, depending on the normality of distribution, we conducted either a Pearson's correlation analysis or Kendall-Tau beta's correlation analysis on significant continuous factors that influenced salinity and fecal coliform to further understand their effects. For this research, we investigated these 4 main groups of factors. Through general linear modeling and correlation analysis, we determined whether temporal, geographical, environmental, and anthropogenic factors influenced salinity levels in the May River, South Carolina. Parameters that are in yellow were not included in our general linear models due to lack of data or how they are measured. However, previous literature and research suggests that they are also significantly influenced salinity levels. I would like to note the exception is population, which we did include in our correlation analysis to better understand how population growth affects salinity levels. We divided the data into 2 separate models. The first on top ranges from 1999 to 2017. It includes the 7 original sampling stations. However, it does not include rainfall data as those data were not available before 2009. Our second model ranges from 2009 to 2017 and includes data from all 11 stations and the rainfall data. For the first model, all factors significantly influenced salinity levels at a 0.05 level. And so this p-value is below 0.05. This model also explains approximately 41% of the variance seen in salinity levels. The factor that influences salinity the most is year, as determined by this partial n-square value or effect size, while the factor that influences the least is lunar cycle, with this partial n-square value being 0.006. For our second model, all factors except lunar cycle significantly influenced salinity levels. This model explained about 59% of the variance, with year influencing salinity the most and tidal cycle influencing salinity levels the least. As determined by the models, geographic distance from the mouth of the river significantly influenced salinity levels in the May River. This figure shows average salinity levels from 1999 to 2017 in the top figure and 2009 to 2017 in the bottom figure. And so stations that do not share a letter were statistically significant or statistically different from each other. As you can see, there's a large spatial variation moving from the headwaters located here at station 1919 to the mouth of the river located here at station 1912. With those stations along the headwaters having the lowest average salinity compared to those stations located closer to the mouth. For our correlation analysis, when data were not normally distributed, we conducted a Kendall-Tau beta correlation test. And so this table shows the results of our correlation tests examining year versus standard error of salinity, which we used as a metric of variability, and oceanic Nino index and salinity. And oceanic Nino index is used as an indice for El Nino Southern Oscillation. The results show that variability has significantly increased in the headwaters of the May River. And so station 1919, located closest to the headwaters, was significantly and positively correlated with standard error, meaning that variability has increased in the headwaters. Additionally, these results show that oceanic Nino index was negatively correlated with salinity at the mouth of the river. So we can see that there's significant values here at the stations located closest to the mouth. These figures provide visual reference for the results into the correlation analysis conducted on year and salinity's standard error. Moving from the top left, which is the headwaters, to the bottom right, which is the mouth of the river, we can see how salinity's variability has changed over time. As we can see, there's a strong positive correlation at the station closest to the headwaters, or station 1919, as variability has increased from 1999 to 2017. Essentially, what this indicates is that there is a larger salinity fluctuation occurring in the headwaters of the May River over time. Research indicates that this may be attributed to watershed development surrounding the volume-sensitive headwaters. And so as development increases, so does the amount of freshwater input into the estuary, increasing what we call the flashiness of salinity levels. Additionally, since the headwaters are not buffered by more stable, high salinity bodies of water, like the mouth is, they show a greater effect due to freshwater input. And so these figures provide a visual understanding of how El Nino Southern Oscillation influences salinity patterns throughout the May River, South Carolina, moving from the headwaters in the top left to the mouth in the bottom right. Salinity is denoted by the light green dots, while El Nino Southern Oscillation is denoted by the blue and red line, where blue indicates La Nina episodes and red indicates El Nino episodes. As we can see in the headwaters, there's seemingly no clear pattern, and salinity is exhibiting more variability. But as we move toward the mouth of the river, the variability decreases and a clear long-term pattern develops. Looking at station 1912, which was negatively correlated with El Nino Southern Oscillation, we can see a clear cyclical pattern that matches the oceanic Nino index. So we can see that as we have these El Nino events, we see decreases in salinity, whereas we have La Nina events, we see increases in salinity. The stations closer to the mouth are buffered by a more stable body of water, so it's easier to see long-term patterns caused by El Nino Southern Oscillation that are lost in the more variable headwaters. And so this table shows the results of our Pearson's and our Kendall-Tau beta correlations analyses examining population's correlation with salinity and rainfall's correlation with salinity, respectively. For population and salinity values were averaged at each station annually since we only had annual values for population. The results indicate that salinity is negatively correlated with population at the station closest to the headwaters of the May River. So as population increases, salinity decreases. We can also see that throughout the entire estuary, rainfall is negatively correlated with salinity, with the most drastic decreases in negative correlations seen at the stations located closest to the headwaters. To better understand the underlying trends in salinity over the past 20 years, we reduced the signal from season, year, and El Nino Southern Oscillation using a 2-year moving average, since El Nino Southern Oscillation exhibits interannual variability, meaning it occurs every 2 to 7 years. The Kendall-Tau beta correlation analysis, using a Kendall-Tau beta correlation analysis, we can clearly see that salinity has significantly decreased throughout the river since 1999. So you can see these decreases moving from the headwaters up here at station 1919 and throughout the entire river to the mouth at station 1912. Development surrounding the headwaters may be the cause of this decrease, as it leads to increased amounts of freshwater input from the surrounding watershed. This, coupled with climate change, which is projected to increase the intensity and frequency of El Nino episodes and may lead to further decreases in these levels, this coupled with climate change, which is projected to increase the intensity and frequency of El Nino episodes, may lead to further decreases in salinity levels. This has major implications for the estuarine species who rely on specific salinity ranges to survive. So I'll now review the fecal coliform portion of this chapter. And so for the second part of this chapter, we investigated the factors that influence fecal coliform in the May River, South Carolina watershed. Through general linear modeling and correlation analyses, we determined whether these temporal, geographical parameters, environmental parameters, and anthropogenic parameters influenced fecal coliform levels in the May River. All fecal coliform data were log transformed before analyses to try and achieve normality, though sometimes within our correlation analysis, some of the data sets were still not normally distributed. Those parameters that are in yellow were not included in the general linear models due to lack of data or how they were measured. However, previous literature and research suggests that they also significantly influence fecal coliform levels. For the statistical analysis of this data set, we use general linear modeling to determine whether these factors influence fecal coliform levels. We divided the data into 3 separate models. The first ranges from 1999 to 2017 and is seen here on this slide. It includes the 7 original sampling stations, but does not include rainfall as that data were not available before 2009. We can see that all factors except lunar cycle are statistically significant at the 0.05 level, with geographic location influencing fecal coliform levels the most, while season influences them the least. And this model explains about 39% of the variability exhibited in fecal coliform levels in the May River. The second model, seen on top, and the third model, seen on the bottom, both range from 2009 to 2017. The second includes salinity, while the third includes rainfall. And these 2 factors were not included in the same models as they are significantly correlated and exhibit collinearity. The second model, we can see that all factors except season significantly influence fecal coliform levels, with geographic location influencing it the most, and oceanic Niño index influencing it the least. And for the third model, all factors except oceanic Niño index and season influence these fecal coliform levels. And we can see that geographic location, again, influences these levels the most, while rainfall influences these levels the least. And the second model explains about 55% of the variability seen in fecal coliform levels throughout the May River, while the third model explains about 52% of the variability. As determined by the models, geographic distance from the mouth of the river significantly influenced fecal coliform levels in the May River. And so this figure shows the average non-log transformed fecal coliform levels at each station from 1999 to 2017 in the top and 2009 to 2017 on the bottom. Stations that do not share a letter are significantly different from each other. And as we can see, there's a large spatial variation moving from the headwaters here at station 1919 to the mouth of the river seen here at station 1912, with higher average fecal coliform levels being found at the headwaters of the river. The red line indicates 14 MPN per 100 milliliters, which is the level at which South Carolina DHEC restricts shellfish harvesting. As we can see, the stations located closest to the headwaters have exceeded this level. So this table shows the results of our Kendall-Tau beta correlation analysis between salinity in fecal coliform and population in fecal coliform. As we can see, salinity is significantly and negatively correlated with fecal coliform throughout the entire estuary, with the most significant correlations occurring at the headwaters of the May River. So up here at station 1919, this value is larger than the values seen closer to the mouth of the river. Research suggests 2 possible theories for this correlation. The first is that as salinity decreases due to freshwater input from rainfall events, fecal coliform levels increase due to runoff flushing pollutants into the estuary from these rainfall events. The second is that lab and in situ studies have shown that the survivability of fecal coliform bacteria decrease as salinity increases. We can also see that population is positively correlated with fecal coliform levels near the headwaters of the river. The fecal coliform values were averaged to generate an annual value that could be analyzed with population data, which were also measured on an annual basis. And so this basically indicates that as population increases, fecal coliform levels in the headwaters have also increased. Similar to the salinity analysis, we reduced the signals associated with El Nino Southern Oscillation year and season using a 2-year moving average. As we can see from a Kendall-Tau beta analysis, fecal coliform levels have significantly increased at the stations located closest to the headwaters, and actually pretty much throughout the entire estuary, with the most drastic increases occurring in the volume-sensitive headwaters. Furthermore, this figure indicates when stations in the headwaters cross South Carolina DHEC's threshold for shellfish harvesting restrictions. And so we can see this red line indicates this restriction and that these stations closest to the headwaters have actually exceeded this level roughly around 2009. These increases are likely due to development and population growth that has occurred within the watershed over the past 20 years. Many studies have linked both population and land use to fecal coliform levels. And so as development continues, fecal coliform is likely to increase. The volume-sensitive headwaters can act as an early warning sign for degradation that may occur throughout the estuary. And lastly, this issue is further compounded by climate change that leads to increased rainfall events due to the changes in the El Nino Southern Oscillation. And that, due to changes in El Nino Southern Oscillation. I'd like to note that this station, 1912, which is closest to the mouth, has actually seen a significant decrease in fecal coliform levels over the past 20 years. And we hypothesize that this may be a result of increasing sea level rise, which leads to greater flushing closer to the mouth of the estuary. And so this slide presents a visual depiction of salinity in blue and fecal coliform trends over the past 20 years. And as we can see, these two factors mirror each other closely. And as fecal coliform increases, we can see that salinity decreases. And so this figure provides further evidence of the changes occurring in salinity and fecal coliform in the May River estuary and their relationship with each other. I'll now review the research conducted for the third chapter of my thesis, entitled A Broader Understanding, Investigating Changes in Salinity and Fecal Coliform Levels over the Last 2 Decades in the Estuaries of Beaufort County, South Carolina. For this analysis, I utilized salinity and fecal coliform data provided by SCDHEC from their shellfish management areas 15, 16B, 17, 18, 19, and 20. The data were taken during their monthly sampling program and ranged from January 1999 to December 2019. These management areas encompass roughly 460 square kilometers in the region and include numerous estuaries and tidal rivers. The population within Beaufort County, which is in this area, has also drastically increased over the past 20 years, growing from roughly 113,000 residents in 1999 to 192,000 residents in 2019. For this research, we only included stations that had been continuously sampled from 1999 to December 2019 to ensure that a complete comparison amongst stations could be made. This resulted in a total of 107 stations included in the analysis out of the 145 stations that are established across the 6 shellfish management areas. Additionally, for this research, we employed the same types of analysis as seen in the previous chapter. Since we determined that El Nino Southern Oscillation was a significant long-term factor for both salinity and fecal coliform, we reduced its signal using a 2-year moving average, revealing a long-term trend in both parameters. This map depicts the Kendall-Tau beta correlation coefficient for salinity at each of its stations included in the analysis. And so stations that have a negative coefficient indicate that there was a decrease in salinity over the past 20 years, while stations that have a blue dot indicate a positive coefficient, which means that there's been an increase in salinity over the past 20 years. Those stations in black showed no significant change over time. From this map, we can see that stations that are located closer to the headwaters of the estuaries or farther inland experience the most significant decreases in salinity, which aligns with the results we saw in the May River analysis. In this map, we can also see significant increases in salinity at stations located closer to the Atlantic Ocean. We hypothesize that this is a result of sea level rise and increased salinity intrusion occurring within these estuaries. And in both cases, significant changes in salinity may lead to negative impacts on species distributions throughout the estuaries, as well as their survival. To better understand which bodies of water have undergone the most significant decreases in salinity, we averaged the significant tau beta correlation values for each body of water located within the management areas. Out of the 33 bodies of water that were assessed in the study, 27 underwent significant decreases in salinity. These are the 10 estuaries that experienced the most significant decreases in salinity. And so our analysis revealed that the Wright River, the New River, Whale Branch Creek, the Ocoee River, the Cooper River, Whale Branch Boyd Creek, the May River, Brickyard Creek, McCallie's Creek, and Uhaw Creek all underwent these significant decreases, with the lowest average salinity for 2019 being found along the Wright River. And so this map depicts the Kendall-Tau beta correlation coefficients for fecal coliform levels at each station included in the analysis. Again, so stations that have a red dot indicate a positive coefficient, meaning that there's been an increase in fecal coliform levels from 1999 to 2019, while stations that are blue have a negative coefficient, indicating a decrease in these fecal coliform levels over the past 20 years. Stations that are in black showed no significant change in fecal coliform levels over this time period. From this map, we can see that the stations located closer to the headwaters and further inland all experienced significant increases in fecal coliform levels over the past 20 years, which aligns with the results that we saw in the May River analysis. In this map, we can also see significant decreases occurring at stations located closer to the mouths of estuaries or closer to the Atlantic Ocean. We hypothesize that this increase in fecal coliform seen in these headwaters and inland are a result of increased watershed development and increased population growth that have occurred within the region over the past 20 years. Conversely, it's hypothesized that the decreases in fecal coliform are a result of the station's location close to the Atlantic Ocean or the mouth of the rivers and estuaries, as they're buffered by larger, more stable bodies of water and experienced increased flushing. To better understand which bodies of water have undergone the most significant increases in fecal coliform, we averaged the significant tau beta values for each body of water located within all the management areas. Out of the 33 bodies of water assessed in this study, 14 underwent significant increases in fecal coliform. These are the 10 estuaries that experienced the most significant increases in fecal coliform. And so our analysis revealed that Habersham Creek, Ocoee River, Uhaw Creek, Whale Branch Creek, the New River, the Wright River, Hazard Creek, the Broad River, Jarvis Creek, and the May River all underwent these significant increases, with the highest average fecal coliform levels for 2019 being found along the Ocoee River. I'd also like to highlight that 6 of these bodies of water are the same bodies of water that experienced some of the largest decreases seen in salinity, highlighting this overlap in these estuaries that are undergoing significant changes. So the next steps for this research should be to dive deeper into understanding the trends presented in this chapter. Understanding the driving mechanisms behind salinity and fecal coliform patterns seen in these estuaries is an integral step in making better informed policy decisions. In addition to this trend analysis conducted for this section, we are hoping to investigate and model the effects of geographical distance from the Atlantic Ocean and land use land cover have on these patterns. This will likely be done through geographic information systems analysis of land classifications surrounding each shellfish monitoring station. The data will be analyzed using a regression analysis. This information will provide deeper context that can be used for future policy decisions. The theme that has been underscored in this research presented is the importance of sustainable development and the conversion of forested land, or conservation of forested land. In the first portion of this research, it was stated that the May River watershed has lost approximately 11.5% of its forested land since the early 1990s. With the loss of this land, hydrological benefits like increased transpiration, or the process by which water vapor is released into the atmosphere by plants, and rainfall interception are lost, leading to increased amounts of runoffs in the estuaries. An example of this being a individual large oak tree can transpire up to 151,000 liters of water per year, using water that would have ended up in the estuary as runoff had the tree not been planted. Additionally, research conducted in North Carolina examined the role of forested land in residential areas and found that tree cover can reduce stormwater runoff by 9 to 21%. And so these figures right here show what happens when you get a loss of forested land. And so as you can see, in land that is completely forested, we have 40% evapotranspiration, only 10% runoff, and 50% groundwater infiltration. But as we start to increase, we can see a loss in evapotranspiration and groundwater infiltration and an increase in runoff. And this is seen all the way up to 75 to 100% impervious cover, where we see very little groundwater infiltration, very little evapotranspiration, but a large amount of runoff. Forest and land restoration is an integral step in improving the health of Beaufort's estuaries. Through policy efforts, local municipalities can start addressing the issue. It's recommended that municipalities investigate means of increasing forested land within the region. An example of this being the Trees to Offset Stormwater program conducted in 12 different cities across the United States. All successfully implemented the use of native trees to help reduce the amount of runoff that not only enter nearby bodies of water, but cause pervasive flooding issues. And this program is developed by the Green Infrastructure Center, and it helped develop community forestry programs in these regions to mitigate the impacts of stormwater runoff in the affected communities. And so we want to look at native trees that we can plant locally to help increase forested land. Research conducted by the city of Charleston found that their trees intercept 28.3 million gallons of runoff annually. They published a table of 10 different types of native trees found within the area and their rainfall interception, which is a measure of canopy cover fraction, and accounts for the tree's capture and storage of rainfall. And this includes evapotranspiration and storage into root systems. And it found that live oaks account for 41% of this rainfall interception. And this table can aid in watershed restoration efforts so that municipalities can utilize the most effective native species. Currently, there's a need for policymakers and local municipalities to take action to address fecal coliform pollution that's become a prevalent threat to estuarine health in Beaufort County. Currently, efforts by the town of Bluffton to mitigate this problem include their septic to sewer conversion program, their septic tank maintenance program, and their microbial source tracking program, which helps understand where fecal pollution is stemming from. However, successful stormwater management requires collaboration amongst regional partners. Locally, 9 jurisdictions listed here have created the Southern Low Country or SoLoCo Regional Board. This board, along with local stakeholders and a consultant team, created the Southern Low Country Design Manual and developed a model ordinance that local municipalities can adopt to address development. This ordinance aims to protect the environment and ensure that point and non-point source pollution does not increase. The model ordinance establishes a set of provisions to regulate post-construction stormwater runoff rates, volumes, and pollutant loads on development sites. The ordinance has strict regulations on when and how developers should address stormwater in their plan. Furthermore, the design manual has guidelines for implementing best management practices, from creating rain harvesting systems to protecting forested land to using permeable pavements in construction. This guide lists how each of these practices may be used and what type of property they would be best suited for. Unfortunately, only one jurisdiction has actually instated this ordinance, which is Beaufort County. The town of Bluffton has brought it before council and it was tabled, and the 7 remaining jurisdictions in SoLoCo have yet to vote on it. Therefore, it is the recommendation that first and foremost, each jurisdiction pass the model ordinance so that stormwater management may become a region-wide effort. I'd also recommend addressing the loss of forested land through programs like Trees to Offset Stormwater program that was discussed earlier, or incorporating forested land into stormwater utility fees or SWU fees. Currently, SWU fees account for impervious surfaces on our property, but by providing incentives like lowering SWU fees for planting or conserving native trees on our property, we can hopefully mitigate stormwater runoff through the maintenance of forested lands. I would also recommend that the other local municipalities instate their own septic to sewer conversion program and microbial source tracking program using the methods modeled by the town of Bluffton. This would address the human sources of fecal pollution, as well as pinpoint other sources of pollution like bird, wildlife, or domestic animals. And lastly, none of these efforts will come easily if there's not the political and local support for them. It is my recommendation that education outreach efforts are expanded to ensure the public's awareness of this problem. This can be done through traditional means like town hall meetings or public speaking events, or the outreach can be incorporated into municipality hosted events like marsh cleanups, rain barrel construction events, and more. This effort can be shared amongst regional partners and like local businesses and nonprofit organizations as well. And with that, so from this analysis, it is clear that watershed development threatens estuarine health throughout Beaufort County. This has likely led to the decreases seen in the salinity in the headwaters of the estuaries, as well as the increases in fecal coliform seen throughout these bodies of water. Furthermore, climate change may be compounding these changes as it leads to increasing and more frequent and intense El Nino episodes. In light of these changes, there's a pressing need for regional cooperation amongst municipalities to address development, manage stormwater, and conserve and restore forested lands. So with that, none of this research would have been achieved alone. First and foremost, I'd like to thank my research advisor, Dr. Montie, who has mentored and guided me since I joined his lab in 2017. I would like to also thank my committee members, Dr. Vulava, Dr. Nowlin, and Kim Jones for their support and advisement throughout this project. I extend my thanks to Dr. Alan Warren for his help on this project, as well as collaborating as a co-author on the first manuscript. I would like to thank my lab managers, Aga and Alyssa, whose constant support and advice were immeasurable. Thank you to WAPAC, who helped bring this project to fruition. And thank you to Mike Pearson and SCD-HEC Shellfish Monitoring Program for their data collection and management. A very big thanks to both the EVSS and MPA departments, as well as my coworkers at the Office of Research and Grants Administration. And lastly, but definitely not least, thank you so much to the family and friends who, if I listed here on this slide, I would be here all day. But especially to my parents and my husband for their constant love and support. It means the world to me. As a note, I would also like to extend my gratitude to the town of Bluffton, Beaufort County, and the Spring Island Trust who helped fund this research. And with that, I open it up to questions.
Perfect job. So for anyone who has questions, you're able to put them in the chat, or you're able to speak up just widely here. And then Eric or Vijay can help monitor if that sounds good.
Sure, that sounds good. Does anybody have questions for Jamileh?
I have a question. I'll jump in if that's okay. Yeah. Congratulations, Jamileh. This was just an amazing project, and I learned a lot from your research here. And I want to underscore that publishing during a master's program is really hard to do, and you should be commended greatly for that effort. And so I look forward to seeing what you do from here. And that relates to my question, which is, what are you going to do from here? I mean, you have, you listed your future directions of this research, but literally, what are your plans from here, and where are you going to take these skills that you learned in the program?
I love that question. So I'm going to take a week to decompress. But after that, I plan on staying in Dr. Montie's lab for the summer and working on the second manuscript based off the second chapter. The second chapter was really exploratory in nature, and so we really want to expand that research into using GIS and geographic location to really pinpoint these spatial variations and long-term variations seen in salinity and fecal coliform trends. But in the future, I hope to work at an organization, either government or nonprofit, that works at the nexus of policy and scientific research, using my MPA degree as well. This will hopefully help with science translation into things that can be used for policy, making better informed policy. I'm really passionate about that. So that's my next steps.
Excellent plan, according to me, that's for sure. And congratulations to Dr. Montie and Dr. Vulava and the rest of your committee as well. So thanks for for working with Jamileh during this time with this work.
Yeah, Jamileh gives us hope in the next generation.
That's a lot on my shoulders, Dr. Montie.
I know. Was there any more questions for Jamileh?
I have a question. So I asked this also to Haley, but I think it's very interesting that this concurrent program that is bringing in so many people who have a background in natural science. Your background is very strong in natural science. You have a very research heavy background within Eric's lab, but then your project did delve pretty deeply into that policy side. Your future work delved deeply into the future implications and how to work with collaborative governance, how to work together. So how did this project kind of change your view on resource management and how to effectively implement resource management?
So not only this project, but I think the MPA program really gave me some understanding and insights into this. And Haley had a very similar answer to what I'm going to say, is that I was very research heavy coming into this. And so I was very much, the science shows, why are we not making evidence-based decisions? But that's not how the world works. And so working in collaboration with local stakeholders who bring different perspectives to the table is an integral step. And so I think through this project, especially, and talking with Kim Jones, who has to deal with this on a daily basis, juggling these oftentimes conflicting perspectives to make policy decisions can be a very difficult thing. And it really takes a lot of effort in working closely with the stakeholders to do so. And also valuing opinions of people who might not have that same perspective as you. I think I talked in Annette's class a lot about understanding worldviews, and you really have to understand the worldview of the stakeholders and the people you're working with to make effective policy decisions, or else you won't have local buy-in or political buy-in or will for it.
That was a really good answer, and it piggybacks on my next one. So what were some of the constraints that you experienced with this project as you're working with natural resources and communicating that?
So I've presented policy recommendations, and I'm hoping to present them to various municipalities beyond just this defense. But I think the biggest constraints that we're finding is the political will. I mentioned that SoLoco has not been brought to vote for 7 of the 9 jurisdictions in it. And so I think really working with developers, working with local stakeholders to see that this model ordinance and that this design manual is very worth it to make a regional wide effort in management stormwater. So
thank you. Those were great answers. Great presentation.
Any other questions for Jamileh? Questions? No. I think, Lucy, is that it then? It sounds like the public doesn't have any more questions.
Yes, well, thank you. I wanted to thank you all for coming to Jamileh's defense. And what we're going to do now is ask the public to give one final round of applause to Jamileh, and then we are going to ask them to leave the Zoom meeting and the committee will question her. So, thank you all.
Well done, Jamileh. I look forward to seeing the paperwork that comes through from this. Bye.
thank you everyone so much.
Congratulations, Jamileh. I'm so proud of you.
Thanks, Max.
Good job, Jamileh, Sarah, and Kelly. We love you.
Jamileh's mama here. Congratulations, sweetheart.
Thanks, Mom.
Give me just a second. Okay, so I think we're good here. And then Vijay, I'm going to make you the host.
Perfect. Thank you for all the help, Lucy.
Yes, you're absolutely welcome. I'll be available through email or phone if you need me through this, but Jamileh, great job. Really proud of you. And I look forward to seeing all this paperwork. Let me know if you have questions.
Thank you.